The major killer during World War I was not battle injury, but infections. This was a time when medical sciences faced a grave challenge. Infections like pneumonia, gonorrhea, small scratches on the finger that could worsen to sepsis and death. In September 1928, Alexander Fleming, a Scottish microbiologist, returned from a holiday to his laboratory in St. Mary's Hospital, London, and began sorting through the culture plates of Staphylococcus bacteria. That's funny, he's believed to have said, when he saw something that changed the world for good. He saw that one of the culture plates had blue-green mold growing on it, and surrounding the mold was a clear area of absolutely no bacterial growth. Fleming identified the mold as Penicillium notatum and named the mold juice that prevented the bacterial growth as penicillin. He found that this juice was capable of killing many kinds of bacteria, but did not at that time have the resource or chemistry background to isolate this juice, which was very unstable. He published his findings in British Journal of Experimental Pathology, but only briefly mentioned its therapeutic potential. A decade later, Dr. Howard Florey, a professor of pathology at Oxford University, was leafing through the back issues of the British Journal. He found this article and set to work with his colleagues in his well-stocked laboratory. After 2 years of experiments with Dr. Ernst Chain, a chemist colleague, and their team, they had gathered enough evidence to show that penicillin could save lives. However, they faced a new challenge: isolating enough mold juice to treat patients. It took 2000 liters of mold culture fluid to obtain enough pure penicillin to treat a single patient with sepsis. Florey realized that large-scale production of penicillin was not possible in Britain, where the chemical industry at that time was focused completely on the war effort. In 1941, shortly before the United States entered World War II, Florey and his colleague Dr. Norman Heatley flew to the US and resumed their efforts with some crucial effort of American laboratories, scientists, and pharmaceutical companies. They soon discovered more effective penicillin-producing strains and began mass-producing penicillin. Penicillin proved its worth in World War II. In World War I, the death rate from bacterial pneumonia was 18%. In World War II, it fell to less than 1%. The overall effects of this miracle drug led to the Nobel Prize in Physiology and Medicine being awarded to Fleming, Florey, and Chain in 1945. Using similar discovery and production techniques, researchers discovered many other antibiotics in the 1940s and the 1950s. And this is how a mold saved the world for good. Do not forget to share this and follow us.